Welcome to Study Loop.
Antibody production in B cells. So basically, we know that antibodies are proteins, so that they're going to follow the central dogma, which basically states that DNA is transcribed into RNA, which is then translated into protein. So in regards to the specific processes by which antibodies are produced, we have the DNA. So very B cells are going to be produced that are very specific for a specific antigen, but they come from a main stem cell. So we have DNA that contains all of the possible combinations of mRNA that can be translated into a specific type of antibody that can detect a specific type of antigen. So the DNA is selected for through an enzyme called recombinase, and it chooses a very specific combination of genes that will produce one very specific antibody for one specific antigen, which is then spliced as shown here and then translated into protein. So if we go down the list and see which statement is true, A states that it starts with RNA processing and then goes to DNA arrangement. So this already contradicts the central dogma. DNA arrangement occurs first via the enzyme recombinase before it is transcribed and then spliced. So A is incorrect. B states that it's first transcription. So transcription right here. RNA processing would be the splicing. But then it states that DNA arrangement comes after that. DNA arrangement is the very first thing that happens. So therefore, we can eliminate B. C states DNA arrangement, RNA processing, and transcription. So transcription occurs before splicing, which is RNA processing. So we could eliminate C. And so D would be the correct answer. So it starts with DNA arrangement, the recombination, then to transcription, RNA processing, which is splicing, translation, and then the production of a mature antibody, which is this final protein right here. So therefore, we could correctly say that the answer